demonstration of Elisa. Here we are going to show you the Elisa on TSH. Now as you can see, this is a kit of Elisa. Here it is TSH kit. This comes in a whole packing. Under it, when we open it, this is a material of the procedure. This is a packing of the plate which is already being explained in the theoretical portion. As you can see, this is a plate on which we are going to show you the demonstration of Elisa. This is a coated wells, 96 coated wells here. This is a washing concentrate of the kit. This is the TMB substrate which is we are going to use. In this we see this is a TSH conjugate. Here are, as you can see, here are all the controls which you can see here. These are different controls which we have marked as A, B, C, D, E. One thing which has remained here is this is a stop solution which is we are going to use at the end to stop the reaction. So now we are going to start with the procedure. This is the samples which we have kept already, which are we are going to measure the TSH levels in the samples serum, which we have already done the centrifuge. Serums have been kept with the serum in the vials. So these are the micro tips which we will be using. These are the micro pipettes which are used. Hence we are going to start forward the procedure. Now we are starting the first step of our TSH diagnosis. Here we have taken out the 96 well coated strip in which it has been already, the wells are already coated with TSH antibodies. Here we have taken out the 8 wells. As you can see over here, these are the 8 wells in which the test will be done. For this we will be taking 5 controls and the rest 3 wells will be taken for the sample testing. This is a control A which we are taking, first control. 50 microliter of this control A will be done in the put into the well, first well. As you can see. Then we will discard the tip and we will take a new tip for it. New tip. Every time we have to discard a tip. Now here this is a control B which we will take in second well. Discarding of the tip, every time we use a new tip. This is a hazardous process, potentially hazardous, so we have to be in very precaution while doing all these steps. This is the control C. We are taking this control C and putting it in the third number well. This is a discarding of tips in 1% sodium hypochlorite. Now we are taking this control D. D control is being put into the fourth well. This is control D, E, which we will take as a fifth control and we will put into the fifth well. Microliter of each is being put into the wells. Now I am taking the sample of serum to be tested. This we have kept at the side so that we don't get the mix up of the vials, serums. This is a sample number 2 which we are taking. Again the pipette is being the tips have been changed after each step. Now in the second 7th well this is added. Sample 2 is being kept at the side. Now we are again taking a new tip and a new sample which has to be tested. This is the third sample serum which has been taken and added to the strip, this micro coated strip, which is already been coated with TSH antibodies. The principle of finding a human TSH level is that the TSH will be sandwiched between two TSH antibodies present in this whole reactions. Now we are taking step 2, conjugate reagent of TSH. 10 microliter each will be added by this pipette and it will be added to each well. 10 microliter. This is a TSH conjugate. As you can see here, very finely this work has to be done. Now the addition of the conjugate has been done and this has to be incubated for 60 minutes and we will keep it in the room at room temperature for 1 hour and we will do the third step after the completion of 1 hour. Now as you can see, we have taken out our strips after the incubation of 60 minutes from the incubator. This has been removed out. Now what we are going to do, the washing of these wells. The washing will be done 3 times and it is done with this solution, washer solution, which is being provided with the kit, but it is diluted with the distilled water in the ratio of 1 ml buffer and 19 ml distilled water. So this is a washing solution. Now we will do the washing.
We discard.
We have discarded the solution of the wells and now we are going to do the washing, taking 300 microliter of wash solution for each well. See, we are taking the marking at the 300 microliter. This we will take from the wash solution. In each well, we are going to fill up these with wash solution. This is the first washing. Now we will again discard the water and we will do the second round of washing. This is the second round of washing. After adding the wash solution, we will again discard it And now again the third round of washing will be done in the similar way. This is the third round of washing completed. This washing is being discarded, wash solution. Now we will dry this on the tissue paper. We will completely dry the strip. By tapping onto the tissue paper, we have dried this coated wells. And now we will proceed further. We will do the next step by adding of 100 microliter of TNB substrate to it. "100 microliter of TNB substrate will be added to each of these wells. Now again we have set our pipette to 100 microliters. As you can see here, this is 100 microliter pipette set, and we are going to use the substrate solution. This is TNB substrate which we will go add to all the wells. Each well is being added with this TNB substrate. Now we will again incubate these wells for 15 minutes in room temperature. We will keep it in the incubator at the room temperature. After 15 minutes, now we are going to add the 50 microliter of stop solution into each of these wells. This is a stop solution for TSH. We are going to use it for 50 microliter for each. After adding the stop solution, we will again wait and shake it gently, and we will wait 10 minutes, 15 minutes again, then we are going to read it. We will incubate it again at 37-degree room temperature for 15 minutes. after 15 minutes we have taken out the strips, and now we are going to read it in the ELISA reader. We will put it into the reader for the readings to come. We will set the reader according to the protocol, and we will take out the readings. In the end of this ELISA session, I would like to give thanks to Dr. Pradeep and Dr. Preeti for my supervising, and Mr. Vishnu for being my technical support. Thank you.